Welcome class to this lecture. In this video lecture, we are going to cover following headings: introduction, definition of acetylcholine, synthesis and release of acetylcholine, mechanism of action of acetylcholine, its site of synthesis and its physiological effect on the human body, and pharmacology of acetylcholine. Before going to the acetylcholine, firstly, we should understand what is neurotransmitter. Neurotransmitter are the substances which neuron produces to use communicate with other neurons and the target tissue in the process of synaptic transmission. It's called neurotransmission. There are more than 40 neurotransmitters in the human body. Some examples are given below. Acetylcholine, its short form is ACH. Norepinephrine, dopamine, GABA, gamma aminobutyric acid, glutamate, serotonin, histamine. Categorization of neurotransmitter. On the basis of their action, it is classified into excitatory, inhibitory, and neuromodulator and neurohormone category. Examples of excitatory neurotransmitters: glutamate, acetylcholine, histamine, dopamine, norepinephrine. Norepinephrine is also known as noradrenaline, and epinephrine is also known as adrenaline. Inhibitory neurotransmitters are serotonin, dopamine, and GABA. Neuromodulators are dopamine, serotonin, acetylcholine, histamine, and norepinephrine. Neurohormones are oxytocin and vasopressin. Oxytocin is released from the hypothalamus pituitary gland, and vasopressin is also called antidiuretic hormone. In addition to the above classification, neurotransmitter can also be classified into the following classes: amino acid derivative. Example is GABA and glutamate, monoamines, serotonin and histamine. Catecholamines. Example is dopamine, norepinephrine and epinephrine. Now we move to the acetylcholine. Acetylcholine is a excitatory neurotransmitter. It means it's a chemical messenger that helps nerves to communicate with each other and with muscles and glands. It is synthesized within the cholinergic neurons from two key components, acetyl coenzyme A and choline. Acetylcholine acts on the two primary types of receptor, muscarinic and nicotinic. Muscarinic receptors are GPCR type of receptor. Its full form is G protein coupled receptor. It is found on the various tissues, including smooth muscles, heart, and certain glands. Nicotinic receptors are the ligand-gated ion channel receptor found at neuromuscular junction and autonomic ganglia. Acetylcholine perform various tasks in the human body. It regulates the heart rate, control the muscles contraction, and influences the glandular secretion. The deficiency of acetylcholine can cause paralysis in the human beings. So it is used for treatment of paralysis. For example, citcoline injection, it contains the acetylcholine analog. In autonomic ganglia, it plays a major role in mediating synaptic transmission, and it ensures the smooth functioning of nervous system. To avoid the constant stimulation, acetylcholine effects are terminated through the enzymatic degradation by acetylcholine esterase. This enzyme is present at the synaptic cleft, and acetylcholine esterase breaks the acetylcholine into acetate and choline, and choline is retransported back to the neuron for reuse for synthesis of acetylcholine. Cholinergic agonist example is physostigmine, which enhances the acetylcholine actions, and the antagonist example is atropine, which blocks the action of acetylcholine. Acetylcholine esterase inhibitors are used in treatment of Alzheimer's disease, and where the boosting acetylcholine levels can improve the cognitive function like learning and decision making skills. Here in this image, you can see Acetylcholine is made from the choline and acetyl-CoA, and it stores in the vesicles in the synaptic cleft, and it rapidly broken down by the enzyme acetylcholine esterase. Choline is transported back into the axon terminal and used to make more acetylcholine, and this cycles moves on. This is postsynaptic cell, and this is acetylcholine esterase, axon terminal. This is acetyl-CoA, and this is synaptic vesicles where the acetylcholine stores. In the cholinergic neuron, acetylcholine is synthesized from the choline and acetyl-CoA, and this secretion is activated by the choline acetyl transferase. This is chemical structure of choline, and this is acetyl-CoA, and this reaction results formation of acetylcholine in the presence of choline acetyl esterase enzyme. By inhibiting the activity of choline acetyl transferase enzyme, we can stop this biological pathway. Here in this chart, you can see the precursor of various neurotransmitter and their site of synthesis. Acetylcholine is synthesized by the choline in central nervous system and parasympathetic nerves, and serotonin 5-hydroxytryptamine 5-st. It is synthesized by tryptophan in central nervous system by chromaffin cells and gut and enteric cells. GABA is synthesized by glutamate in the central nervous system. Histamine is synthesized by histidine in hypothalamus. Norepinephrine is synthesized by tyrosine, and dopamine is synthesized by tyrosine, and nitric oxide is synthesized by arginine in central nervous system and gastrointestinal tract. So here we have completed the pharmacology of acetylcholine. It is important for exam point of view. In past years, it has asked many times. So make note of this topic, and thanks for watching this lecture.